Hi everybody, welcome back for day 2. We have been talking about proteome informatics, and the subject last week was all about identification. How do we go about identifying peptides from tandem mass spectra? How do we figure out which tandem mass spectra have been successfully identified? And how do we infer a set of proteins to explain the peptides we've observed? So that was a lot of material to absorb. I hope you were able to make use of seeing the video again on YouTube, because I know a lot of people were feeling a little glazed over as we hit about the hour mark. And that was, we still have another 8 minutes to go. I'm going to try to keep things nice and compact today, but again, the field is rather complex, and I hope to make sure that by the end of this lecture, all of you know, for example, how isobaric or iTRAQ labeling differs from something like total labeling through SILAC methods. So there are lots and lots of places where quantitation within proteomics has evolved over time. So I'm trying to encapsulate all of those in one lecture. All right, so let's move ahead to the first slide. We're going to be talking about the subject of proteomic quantitation. We're going to start with kind of a basic label-free method called spectral counting. Some people already perceive this as kind of an old technique, but the reality is that you can do an awful lot that's useful with spectral counting. From there, we're going to talk about another way to evaluate the same data via extracted ion chromatograms, integrating areas for chromatograms. Then we'll talk about constitutive or isobaric labeling techniques, the use of heavy isotopes or molecules that are designed to make different isotopic labels for particular samples. And finally, selected reaction monitoring. This is a method that we frequently use to do a confirmation of differences detected in a shotgun data set. Remember, everything we talked about last week was shotgunning, about doing a discovery experiment where you're asking what are all the different proteins and peptides that we can detect in this mixture. So, having spent a little time on the left side of this graph last week, we were working on database search and on FDR assessment and protein inference. Today we're going to move over to the right and start talking about techniques that apply in both proteomics and in metabolomics, especially as we move to the latter part of the lecture. So there are lots of places we could be talking about proteomics, but in this case we're going to be focusing on techniques like peak integration and spectral counting particularly. So, spectral counting. This is a label-free technique intended to ask a very broad question, a very broad quantitative question. If your research needs to know, does this protein's level increase by 20% or by 12% between a pair of cohorts? That's a very narrow kind of question to ask. It's a very specific one, and you wouldn't want to go after something like that with spectral counting. On the other hand, if you had cohorts of this type and cohorts of that type, and you want to know which proteins change between cohorts, spectral counting is a really good option for you. So if you're trying to ask what changed, you're going to have different requirements of an experiment than if you're asking by how much did this change. Do people see that distinction? Okay. So the way that this label-free technique works is that it envisions all the different peptides in the sample as competing for attention. Imagine for a moment that you're a teacher in a classroom, and you have students that are each trying to get your attention in different ways, right? You ask a question at the front of the class, and there's one person in the class, there's inevitably one person in the class who's always like, ooh, 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 that's me, that's me. But imagine that you have another student across the class who's much more introverted than that student, but very, very smart, right? That person is not likely to be clamoring with their hand up in the air, but they're a person who could illustrate that she or he understands that answer too, right? So you can imagine that this clamoring for attention can cause some ions to get a disproportionate amount of attention. But in this case, some proteins get a disproportionate amount of attention. So, if you have a protein that changes in concentration between two similar backgrounds, you would expect that when that protein is more concentrated relative to the mean, it's going to get more attention from the mass spec, which is to say more peptides are going to be analyzed from that protein. More tandem mass spectra will be produced for their peptides for that protein. So more concentrated proteins will in effect match to larger numbers of tandem mass spectra. If the same protein is observed in different samples, the number of spectra that match to it are predictive of its relative concentration. So if there's twice as much of a protein in one cohort as in the other cohort, and they have otherwise similar compositions, you can detect that the number of spectra has jumped in one cohort versus the other, and learn that that protein has changed. Okay. I want to specify that there's a point about this that a lot of people misunderstand, which is to say that mass spectrometers today use software that's designed to prevent the reacquisition of tandem mass spectra for peptides that it has already seen. Right? So if you've already collected a tandem mass spectrum for a particular peptide, do you need to snapshot it again in order to identify it? No, you've already seen it, you've already identified it. So a lot of people say spectral counting could only work if you allow the machine to just take tandem mass spectra whenever they appear. But this misses a really important point. In spectral counting proteomics, we see that the number of peptides that we observe from a protein will change as a function of concentration. So this protein may have 10 peptides in evidence in the first cohort and 20 peptides in evidence in the second cohort. So it's not just a question of multiple spectra for the same set of peptides, it's rather a different set of peptides being observed every time. All right, so why is this working? Where trypsin cuts is a probabilistic process. You may have 10 tryptic cutting sites in a protein, but they're not all cut with perfect reproducibility. Right? If you use trypsin today and use trypsin tomorrow on exactly the same proteins, which peptides will be released from that protein is probably going to change slightly. So this means that rare products of trypsin digestion are going to show up with greater probability if you've got more copies of that protein to chop. A peptide will compete with others for ionizing protons in nanoelectrospray. So in the process of electrospray, you have some number of protons that you can stick onto these peptides coming off the column. And if some of these peptides ionize really well, which is to say lots of basic residues like arginines and lysines and histidines, they are likely to outcompete peptides that have fewer basic sites, fewer arginines, lysines, and histidines. So lysine and arginine have a story beyond trypsin. They also have a lot to do with the ionizability of peptides. Every proteomic sample contains too many peptides for the mass spectrometer to produce a comprehensive set of MSMS scans. So at this point, you probably should be kind of rolling your eyes at Professor Tabb and saying, no, really, come on. If you shoot one protein in LCM-SMS, shouldn't you be able to see all peptides from that protein in a single experiment? I mean, one protein, how difficult could that be? You might think that. You might think that. Imagine that a protein contains 9 cutting sites for trypsin. Therefore, you have 10 peptides that, in theory at least, should be visible. But you're missing the fact that when you shoot one protein at a time on the mass spec, you also see semi-tryptic peptides that don't have good trypsin cutting sites on either end. You also see oxidation products. You also see charge state variants. So shooting one protein, say BSA, again and again and again on the mass spec is still going to show variability about which ions are selected for tandem mass spectrometry. All right, MS scans may be acquired and yet fail to be identified because a spectrum that's caught at strong signal to noise is a lot more likely to be identified successfully than one that's right at the edge of detection. Do you see that? The stronger the signal, the better your shot of identifying it. As a result, peptides are always in competition for which one is going to get mass spectrometry time and which will be identified. Those from abundant proteins have an advantage. Now these are kind of old data at this point, but I think the point is still very commonplace. You will very frequently find in spectral counting studies tables that look just like this. So each row here starts with a P. These are all Swiss prot protein accessions for proteins in E. coli in this case. We have shot 5 samples that are red 1 through red 5, and 5 samples that are yellow 1 through yellow 5. And we're trying to determine which of these proteins, represented by rows, are changing between these two cohorts. So if you were to see one of these proteins has a huge number across the yellows and relatively low numbers across the reds, that might be some indication that this protein has changed in level. But at the same time, we must recognize there's a lot of variance associated with this. If you look across these columns, we see that in this particular row, we have a high value of 43 and a low value of 33. So that's a lot of internal variance for one cohort. How we deal with data of this sort to come away with decisions about which proteins are changing in concentration is something that a lot of people have taken some time with. There are lots of different statistical tests that have been created for this purpose. But because these are spectral counts, what we have are a bunch of integers to work with. These are counts, not values that are, say, normally distributed. They're generally more like Poisson distributed. It's kind of a mess. So I'm not really going to talk a lot about the statistics on how to do this at the moment, but you should be aware that the question of how you turn a table like this into decisions about which proteins are changing is a complex place to work. All right. Everybody should be ready to define the term chromatogram after hearing this lecture. I'm sorry, that's my obligatory, I like asking questions about this topic warning. Chromatograms are the natural result of chromatography. Does anyone know where the term chromatography comes from? What's chroma? What does that root mean? What's that?
Attaching something that can emit light.
Light?
Color.
Okay, that would be photo. But the
color.
Exactly. Yes, very good. Yes. So yeah, if we were talking about photography, that would be picture writing, right? This is chromatography. So here we're creating a written record of color in this case. So chromatography is a separation of color. A lot of this comes from some of the very earliest experiments in the field of analytical chemistry. Do people still study things like TLC and techniques like that for separating photosynthetic pigments? You know, if you take a fall leaf and look at the distribution of dyes that are that fall within leaves, that they're changing as okay. Well, anyway, that all came out of some of that area of study was one of the very first applications of analytical chemistry, and so chromatography became the study of separations essentially. So in our standard LC MSMS experiments, we are most typically using a chromatography called reversed phase liquid chromatography. This RPLC is giving us a time axis. All right, so as a function of time, we are seeing different peptide ions from a very complex peptide mixture. Everyone's good with that stage? That's our time axis. Now, we could be talking about a total ion chromatogram, which is to ask how much electrical signal are we producing in the ions we can detect as a function of time. This is a combination of all the different peptides that are being detected at any one point in time. So a TIC, a total ion chromatogram, is just a way to say how much signal is reaching the mass spec over as a function of time in an RPLC experiment. I'm being a lot more specific here though. When I get to chromatograms in this context, I'm really talking about extracted ion chromatograms, or XICs. An extracted ion chromatogram is about a particular ion, a particular ion's passage over time the intensity associated with that particular ion is a function of time. So a chromatogram then is a record of how much electrical intensity we're getting from the detector of the mass spec as a function of time. That is a chromatogram. So I think that you can argue lots of things from a chromatogram. It could be that if you're trying to track a particular peptide, that you know it should show up around, say, 30 minutes, maybe around this time too here. But that's not to say that's the only time you're going to observe that m over z value, because there are other peptides with different retention times that also have the same m over z value. And so they could appear at different retention times and still produce a peak of intensity there. So generally speaking, there's the possibility of complications where multiple ions could contribute intensity for a particular mass to charge value. But chromatograms are a very standard way for people to characterize how much of a thing have you got. Chromatogram is a concept that applies whether you're doing metabolomics or proteomics, so everybody should really have that concept. Now, we can make this even more complex. Before, I suggested that we were parking on some particular mass to charge value and simply saying, how much intensity do we have for this now? How much intensity do we have for it at the next instant, or the next instant, or the next instant? But you can think of this as a whole plane of space, right? So imagine that we have, I'm going to use the table for this. Imagine that we have time being forward and back on this table. Now let's imagine m over z as being the width of this table. So we have lots of ions showing at lots of m over z values as a function of time. We might have Mount Everest right here, but we might have a Pikes Peak right over here. These small peaks then are living in this space alongside all those big peaks that represent the really strong signals within this. In this case, we're looking at a 3D volume reconstruction of a very, very narrow slice of mass to charge, maybe 20 mass to charge units, okay? And we're looking as a function of time. In this case, maybe 30 seconds. So instead of looking at the entire run, we focused on this vanishingly small little area of m over z and of intensity. And we're looking at one peak abstracted away from that big space of all the peaks we saw in the course of an LCM-SMS experiment. So in this case, we're able to do two kinds of corrections here. One is that because we are looking at all of the m over z values within a 20 m over z space, we can see that some intensity is visible even a little bit off the center of where we expected this peak to be on m over z space. And we can see that this peak is not just appearing in one MS scan and then vanishing. It's rising rapidly and then trailing off as a function of time. So MaxQuant is one of the major tools used in South Africa and in Europe and at a lot of places in the United States for that matter. Came out of the Matthias Mann Lab. And this software is capable of using this 3D model of a particular feature in this chromatographic space to associate a particular peptide that's been identified from tandem mass spectrometry with an intensity that's been integrated in both m over z and retention time space. Generally speaking, the software is not used to compute a volume in this way, but rather to fine-tune what mass to charge value it will pay attention to represent this particular peptide. So MaxQuant is a very popular tool out there. I want you to keep in mind that what we are measuring in tandem mass spectrometry is peptides, not proteins. So how the software is able to deal with lots of peptides all being quantified in this way and wrap that up into a single protein intensity measurement is actually kind of complex and important. All right. So now that we've talked about chromatograms, it's time for us to talk about labeling. So most generally, you will find three different elements that are used to introduce a heavy mass into something. C, N, and O. Okay? So C is carbon, N is nitrogen, O is oxygen. When people do heavy oxygen labeling, it means that they're usually introducing heavy water, for example, in a digest of a protein, which means that when you use trypsin on a protein and it breaks a peptide bond, it's not sticking on an ordinary OH, it's sticking on an 18O and H. So you can see why that would be 2 Daltons higher than if you used ordinary water for this digestion. That's an example of how you can incorporate a label into existing proteins. Okay, so the idea here is that those extra neutrons we're adding do not change the biochemical behavior of that peptide. So imagine that you have a labeled sample and an unlabeled sample. One you digested in heavy water, one you digested in ordinary water. You would expect that a given peptide from these two samples that appears in both heavy and light forms will elute at the same retention time. But they're going to differ in the mass to charge value they inhabit. Do you see that? Okay, so we expect that the biochemical property of hydrophobicity is not changing. The retention time does not change. But the mass difference is discernible easily by today's mass spectrometry equipment. All right. When you are using a labeled standard, you are able to make statements about the quantitation of a particular protein. You will frequently find proteomics people saying, we have absolute quantitation values on this particular protein because we know exactly how much labeled version we put in there. But I would just point out then that they are taking their measured chromatogram for unlabeled peptides and comparing it to a heavy labeled version as well, which is already a relative process. So I would say that the term absolute quant gets thrown around very loosely in our field, when in fact it's all relative. All right. Now, I would note that there are lots of things that could go deeply wrong. Imagine that when you did your heavy digestion, a heavy water digestion of your protein, that you had only 90% heavy water and 10% light water. That means that you'd have a 10% chance every time a digestion took place of incorporating an ordinary OH rather than a heavy OH. You see why that would cause a problem? Now you're adding a little bit of signal to the low intensity one with 1 in 10 of the digestion processes. So having incomplete labeling can distort the kinds of isotopic packets that we see for these proteins and can greatly complicate your ability to pull a meaningful ratio out of it. All right. So back when I was in grad school, metabolic labeling was kind of the standard way to go. And remember I said that C, N, and O were the most common ways that we did this. So let's imagine you're growing a microbe on a dish. And let's further imagine that this microbe has no ability to grab carbon from something like CO2, right? So its carbon source on this plate of media is whatever sugar you put in there, in all general, if I'm being very general about it. So if I ensure that the only carbon source available to this microbe is heavy labeled glucose, then over generations and generations of these microbes growing, the fraction of all of the carbon in these bugs that is comprised of heavy carbon is going to rise and rise and rise, and it's going to get really up in the high 90s of how much of its overall carbon comes from this labeled source. So that's an example of metabolic labeling or constituitive isotopic labeling. All right, some people will do that with nitrogen as well. So again, if you're not able to incorporate nitrogen from the atmosphere, then you're getting your nitrogen from somewhere else. Being able to change that to a heavy labeled version over time is very valuable. However, while this is great for bugs, it's not so great if you're dealing with mammals, right? How many humans that you know would be willing to have to be fully labeled for all of their carbon 12s to be replaced with carbon 13s, or all of their nitrogen 14s to be replaced with nitrogen 15s? It's not a very popular option. That said, people have done experiments where they labeled a rat, for example, they found a type of algae that you could, with enough starvation, convince rats to eat, such that over the space of months, these rats began having almost entirely labeled nitrogens or carbons. I've forgotten which one. I think that Wu paper from 2004 talks about that experiment. Rats apparently don't like eating mold. I don't know why, but it's true. So, not very nice experiments. So people have argued that that's not a good way to go about dealing with mammalian samples. So there was a big paper on the incorporation of labeled amino acids. You remember my mentioning the Matthias Mann laboratory in connection with the MaxQuant software a couple slides ago. They also put together an approach called super SILAC. So SILAC is a stable isotope labeling technique. We used to use SILAC to describe this very first method, but in time, this SILAC method has come to mean that we buy amino acids that are labeled. So if you have a cell line that is only able to take its lysine and arginine from the environment it's growing in, you can provide it these amino acids so that all of their lysines and all of their arginines carry an extra neutron or two. So this incorporation of labeled amino acids is really good, even in mammalian systems, if you're working in cell culture. So you'll frequently see that will go on. Now the super SILAC thing was the argument that you can create labeled human proteins from SILAC and then compare unlabeled proteins from biological samples with that. There are a lot of problems with that approach, but you should be aware it exists. Then the final approach that we're going to talk about is this isobaric labeling, iTRAQ or TMT approach. This is a very commonly used approach for people who are trying to do their quantitation very carefully in a context where labeling otherwise is not possible. So if you're not able to do a comparison against some cell line model, then you probably would make use of isobaric tagging, and we'll talk about how that comes into play. So how do you translate this labeling approach into useful data? I want you to imagine that we have a cell line model in this case. So we have labeled with N15 enriched media. So one of our bugs in this comparison on the left is heavy, and the other one, the experimental one, is light. So we're going to use this heavy version of the thing growing in a plate as an internal standard. In experiment 1, we mix the peptides from our heavy labeled bug with our unlabeled sample 1, and then we're going to compare the intensities of heavy and light peptide pairs between those two. We use the same internal standard over here in experiment 2. You see that sample 2 is also compared directly with this internal standard. So if you saw that a particular peptide from sample 1 was up by 20% versus the internal standard, and on the right cohort you saw that it was down by 20%, you might argue that sample 1 has this peptide at a much greater fraction than the sample at the right. Do you see how that works? That in one case we were 20% up, in the other case we were 20% down. So we argued that there's much more of it in sample 1 than the other two. And the reason here is that both of these can have this internal standard divided out. Because A gets compared to S and B gets compared to S, we're able to pull S out of that fraction to say, how does A compare to B directly? Do people see the math there? Okay. Now, people have used this SILAC approach, one where we label with particular amino acids, to do things like time points. In this case, we're using an unlabeled, sorry, I have to keep this one straight in my mind. We have different kinds of labels available to us for arginines and for lysines. So it's not just a simple split between heavy and light. We also have like medium versions of these. This gives you the ability to combine together data from multiple time points if each of these different labels represents a different time point in your experiment, to do one proteomics experiment that combines together data from multiple time points. Then you can keep track of how much signal comes from each of these cohorts based on which mass to charge value you're getting what chromatographic intensity. Did I make any sense? Yes and no. Maybe that was just a no. That was just a no. All right, we'll try it again. We are using different amounts of different types of labeling to reflect different time points in this study. You could just as easily do this as an A versus B versus C comparison. You know, all the stuff we've been talking to about this before has been a heavy versus unlabeled, basically. In this case, we're allowing for different levels of labeling taking place. So if you were to look at this, you would see in this case we have a 0-minute, a 40-minute, and a 160-minute experiment that are all being run together. And we're able to compare one peptide, in this case LLQ-LVEDR, based on what weight that arginine is contributing. That's only the arginine is differing among these three peptides with the same sequence. If it's got no label, we know that it comes from the 0-minute cohort. If it's 6 neutrons higher, we know that it was the lighter version of the labeling. And if it shows up at 160 minutes, we know that it came from the arginine plus 10 labeling. So in this case, we have 3 different experiments getting all commingled into one LCMS-MS experiment, where each of those time points is differentiable by how many neutrons that peptide is carrying around extra. 0 is the unlabeled version, 6 is the middle level, and then 10 is the heavy labeled version. Obviously, interpreting data like this gets to be a little messy, because in this case, one peptide gets 3 chromatograms, one representing each of these 3 labeling states. So SILAC is very powerful for doing techniques like this, and gets used when people have access to this sort of thing. In South Africa, it's a little harder to get access to heavy labeled elements, but it is feasible. All right. Now this last one I think may be the least accessible, but it's important to know about. iTRAQ and TMT labeling experiments tend to be a little bit more expensive to run than these other routes. Acquiring radioisotopes is frequently expensive, right? So none of these labeling approaches are as cheap as just doing label-free stuff. But the benefit, as I've already alluded to kind of loosely, is that you can run the samples together. So less instrument time, but more expense in creating individual samples. So in an iTRAQ experiment, you do not count on having introduced any label at all into the sample cohorts. So imagine that you have sample cohort 1, sample cohort 2, sample cohort 3, sample cohort 4. These could represent 4 different time points, or they could represent different stimulations, right? So our friend right here just had an experiment with 2 different species combined with 2 different levels of water supply to these 2 species. So that 2-factor experiment translates into 4 cohorts. Did everyone see that part? So think of these as A, B, C, and D. Now I want to run an experiment where I ask, does this protein change among cohorts A, B, C, and D? iTRAQ is really, really good for that. So in iTRAQ, after you do your sample digestions, you've digested the proteins of A, you've digested the proteins of sample B, you've digested the proteins of sample C, and you've digested the proteins of sample D. Now, we want to combine these 4, but we need to be able to tell which sample produced this peptide. So we are going to use an isobaric label. In the case of something like iTRAQ, for example, I might have 4 different reagents that are designed to be used together. If I label the samples, if I label the peptides from cohort A with reagent 1, and the samples from the peptides from cohort B with reagent 2, the peptides from reagent from cohort 3 with the third isobaric reagent, and those from cohort D with the fourth, I'm able to mix those together after the fact and run them together in one fourplex, we would call this. So it might sound like I've lost information. I mixed these samples together, right? But here's the trick. On mass spectrometry, these 4 samples look very, very similar. They have very, very similar mass to charge values. But when I look at them under tandem mass spec, something magical happens. Each of these 4 reagents nominally has the same mass. But when I run them through a tandem mass spec, they each produce what's called a reporter ion. And these reporter ions break off during the process of CID. So we've already talked a lot about CID last week. Remember, we had peptides breaking at peptide bonds to create B ions and Y ions? Adding iTRAQ reagent changes that a bit. It adds a new kind of breakage where reporter ions are released from these peptides. So the peptides that came from cohort A produce the 114 reporter ion. Those that were produced with isobaric reagent number 2 produce the 115 reporter ion at that M over Z value. The third one will produce a reporter ion at 116, and the fourth will produce a reporter ion at 117. Here's the magical bit. After doing an iTRAQ experiment from an individual tandem mass spectrum, you can ask, how much of this peptide came from which cohort? Does everyone get that? I realize a lot of this is really complex, so I hope you'll bear with me on this. When you have a tandem mass spectrum produced in an isobaric reagent experiment, in an iTRAQ experiment or a TMT experiment, you can look at this low mass region of the tandem mass spectrum. Up above this, we've got all kinds of B ions and Y ions going on. That's the same in all kinds of tandem mass spectrometry. But down here in an iTRAQ experiment, these reporter ions allow us to say how much of this peptide came from which cohort. So we see in this case that 115 is rather tall, 117 is second highest, 116 and 114 lag behind that. So here we could say the sample that contributed the most of this peptide was number 2, because isobaric reagent number 2 is the one that's producing most of the fragments there. So here we have reporter ions that are telling us that how much of this peptide is being contributed by which sample. We ran these samples in one MS experiment, in one tandem MS experiment, and the reporter ions are going to tell us how much of each peptide comes from where. Very powerful capability, because we no longer have this requirement that we introduce the label in vivo. You could be working on samples from a human being. You could be working on biopsy stuff, right? You don't have to label the person in order to do this kind of experiment. You're labeling their peptides long after they're out of the person. So this kind of after-the-fact labeling has been incredibly powerful for a huge number of experiments. Okay. So to this point, I've really been talking mostly about shotgun experiments. These are cases where we were trying after the fact to get quantitative information out of a shotgun experiment. Somebody produced a big protein inventory and they wanted to say which proteins changed from one cohort to another. If you want to be much more precise than that, if you want to be able to answer questions about whether or not you can trust a 20% change for some peptide, it's really very important that you step away from shotgun experiments and focus on experimental designs that are targeted at a particular set of peptides. And for that, we have methods like SRM, which we're going to be talking about from this. So why bother with it? The proteins that we generally think of as most interesting in biological experiments are not the top 10 proteins from that sample. Right? If you're looking at blood biomarkers, it's probably not all that interesting to you to know whether the amount of albumin is apparently higher or lower between a bunch of samples. Albumin is not generally the protein that people look to for diagnostic significance. So, interesting proteins tend to be those that are a few layers down. They tend to be proteins that are present at very low concentrations in these samples. So, being able to focus the mass spectrometer's attention on these interesting proteins rather than the grand inventory of them all is very important. Any given shotgun method that's just sampling to see what peptides are there is much less likely to see the same peptide again and again and again. If you try to compare all the peptides seen in one experiment and then shoot the very same sample again in the same instrument, you're going to see a rather different set of peptides every time. This is due to the sampling nature of shotgun proteomics. That limits our ability to make statements about whether peptides are changing or not. If you're able to target a particular peptide for quantitation, you're going to achieve a lot higher sensitivity and generate an intensity profile, a chromatogram, that you can really trust. Generally speaking, if you're doing proteomics experiments in support of your work and you want to go publishing that in a journal, it is not enough to just do a bunch of shotgunning and say, these are the proteins we found to be different, give me my PhD. It doesn't work that way. I'm sad to say, we have found through bitter experience in the proteomics community that just performing a shotgun experiment is not enough for nailing down which proteins really change. We really need another round of experiments to confirm these differences. And for that, a targeted experiment is a very good way to go about confirming a difference that you found in a shotgun method. So for this, we most typically use a rather different kind of instrument. Have people heard of a triple quad? Triple quad? Any mention whatsoever? Okay, I'll give you an introduction. A triple quadrupole instrument is still a mass spectrometer. And what are the three things all mass spectrometers contain?
Ion source.
Ion source, very good, yes.
Mass analyzer.
Mass analyzer is number 2, yes.
And a detector.
And a detector, brilliant, well done. Okay, so in this case, the ion source is shown at the upper left, the detector is shown at the bottom right, and the mass analyzer is this complex of 3 quadrupoles. Okay, the mass analyzer, in this case, is going to have a different kind of function for each of these quadrupoles. The first one is going to say, I'm interested in getting a signal for this particular peptide's mass to charge value. Okay, so we know what m over z this peptide appears at. And so the Q1, the first quadrupole, is set to pass only ions of that particular mass to charge value. Q2, in this case, is set as a collision cell. And a collision cell has one function: blow it up. Whatever is passing into the collision cell is going to have CID caused, and it's going to break apart into peptide, into B and Y ions most typically. And finally, Q3 is going to be set to look for a particular fragment known to be associated with this peptide. So I'm going to give you a quick hypothetical. Imagine that you think these 50 proteins, here's my list, these 50 proteins are likely to be differential between my cohorts. So you've got some hypothesis you're starting with. These 50 proteins are known to produce these 3 peptides each when I analyze them in tandem mass spectrometry. Okay, so we started with 50 proteins of interest, and we've now turned that into 150 peptides of interest. All good so far? Okay. Next, we know that when we see tandem mass spectra of these 150 peptides, we can expect these 3 fragment ions to be observed for each one of these peptides. Okay, so we had 50 proteins, they became 150 peptides, those became 450 transitions. Transitions, that's a really important term. Even if it doesn't appear on the slide here, the term transition is one you should definitely know in connection with targeted mass spectrometry. A transition represents a particular peptide mass to charge value and a particular fragment mass to charge value that together are something you can measure to quantify how much of this peptide is there. Now, why would we bother measuring multiple fragments and multiple peptides for a given protein? We want to have multiple lines of evidence to quantify this particular protein. By having multiple peptides, if we see that 2 of these peptides show a rise by 20%, and 1 of these peptides shows a fall by 60%, we want to know that to give us some estimate of how much assurance we have about this quantitation being valid. So you can imagine that the total targeted proteomic workflow, starting with a peptide mixture, we've already digested it up. We're using liquid chromatography, same as we did with shotgunning. We're still using electrospray ionization. But now the triple quad is screening out all but this particular peptide ion for this moment in time. We are colliding those peptide ions to cause them to dissociate. Then we're checking for this fragment ion, then that fragment ion, then that third fragment ion for that particular peptide. We're able to put together traces that represent the chromatograms for these individual peptides, for the fragments we've seen from those peptides. We're able to integrate them and compare these areas to some sort of reference. A lot of people will buy labeled peptides so that they know exactly how much they've got of this particular experimental peptide. So what do these look like? All right, so when we look at a chromatogram, remember we still have our retention time measure. Remember that when an individual peptide shows up in LC-MS, it tends to show up for around a 30-second interval. So you can see in this case, we have this 30-second interval right around 52 minutes, and we have multiple traces rising and falling together. This is one peptide ion that we are measuring 5 ways, right? We have 5 transitions that we measure for this individual peptide. You can see that they represent the Y7, the Y4, the Y6, the Y3, and the Y5 ions from this particular peptide sequence. We are glad to see that all 5 of these show up together. And I want you to think about why that would be. In the LC retention time for this peptide is common. I mean, that one peptide produces all 5 of those traces. Does everyone see that? These 5 transitions must be correlated together because this peptide, this individual peptide, is producing all of them. So over here, we see that the Y6 ion, this little purple trace, is showing up by itself down here. That's not where the peptide is eluting, because that one peptide, that one fragment shouldn't be seen by itself. It should only be seen in combination with the others. So this is a false lead. This space over here, we see that the Y3 ion is showing up at like 49.5 minutes. Again, it's a false lead. That's not where the peptide is, because all of the fragments should show up at the same time. So the software is able to recognize that they all rise and fall together, and all 5 of them are present. This is the part we're going to use for quantitation. All this other stuff is not useful to us. Doing all right? An individual peptide shows up at one particular time, and all of the fragments that we see for it should show up at the same time as well. Okay, so we've started with chromatograms, and now we've looked at how that would be reported in a different kind of mass spectrometry. Instead of LC-MS shotgun discovery proteomics, we're spending our time looking at targeted proteomics, where we know in advance which peptide sequences we want to quantify. Okay, Skyline is some software that I worked with long ago now. It has rapidly, basically pushed aside lots of other software workflows that exist, and a lot of people use it now. So whether you buy your instruments from Bruker or Shimadzu or Thermo or AB SciEx and any of these companies, many people today will end up using Skyline to design their targeted proteomics experiment and to analyze them. It's a very widely used piece of software. So you can see that it's got kind of a friendly user interface. Over at the left, I realize it's very small, but you can see there's kind of a hierarchy showing up. What we have are different proteins giving rise to different peptides, and those different peptides are being measured via different transitions. So all of that is organized for you at the left edge of this. You can compare peak areas for individual peptides for an individual protein. You can evaluate whether the retention times at which a particular peptide is seen among different experiments is consistent or not across its transitions as well. So it's a rather friendly piece of software to help people design experiments and characterize the data that come back from them. Now, I understand that you've observed calibration curves before, but I just feel like we have to mention it any time the subject of quantitation comes up. A calibration curve happens because our researchers are not interested in mass spectrometric intensity. The mass spec is a tool, right? And in this case, the outcome, the output from using that tool is quantitative information. So ultimately, people want to be able to translate mass spectrometric intensity into something like concentration that they can use. And for that, a calibration curve is a very standard way to go. So we have things like limit of detection. I want you to imagine that you are looking for 50 human proteins, and so you have 450 transitions corresponding to those 50 proteins for 150 peptides that you're going to go looking. But let's be crazy for a minute. The samples we're going to use to look for those human proteins don't have them present at all. Right? Let's say we're looking in yeast for a bunch of human proteins. Well, the human proteins aren't there, so wouldn't it be nice if the mass spectrometer gave you an intensity of 0 for all the peptides you saw for all of those proteins? That would be great, wouldn't it? But that is not reality. The reality is, even if 0 of your protein is present in that sample, the mass spec is probably still going to give you some sort of chromatogram to work from. It may be dismal, it may be full of noise, but there's still some amount of measured intensity for these peptides, even when there's none present. That's not surprising anyone, is it? You've done some experimental work, you've probably already encountered this in some way. Okay, so I would suggest then that a blank sample like this shows you what the amount of intensity would be observed even when 0 is present. Those blanks help you to determine a limit of detection, to say above what level do I need to ascend before I claim that this thing is actually present at all. Now the limits of quantitation are rather different. For limits of quantitation, we're trying to characterize a lower bound and an upper bound on how much of this protein can be present such that we can quantify it accurately. So this is a little strange. We think of this as kind of the area of linear response range. When there's twice as much protein present, you want twice as much of the signal to be present. Everyone good with that? But if you're way down at the very bottom end of sensitivity, it may be that adding twice as much peptide does not double the amount of signal you see. So we need to understand where the errors are very low, where the variation is quite low, such that any additional signal leads to an increase in the response as well. Now, I mentioned that we have not just a lower bound on this, but also an upper bound. I want you to think of the experience you had the most recent time you went to a concert. Has anyone gotten a ticket for a concert that inadvertently got you stuck right next door to the speaker? Never? No one's ever had this experience? Okay. Well, this always seems to happen to me. I get a cheap seat for the concert, and then I find I'm sitting right next to the speaker, and I tell you, I hear nothing for days thereafter. It's a really big problem. This is a problem because our ears, like lots of other detectors, have some limit above which they don't really go very effectively. And even if the sound level is doubled in intensity, to us it all just sounds like a big blat. Well, mass spec detectors are very much the same. If you have too much of the sample there, you're not going to be able to detect that its level goes up or down because all the instrument hears is blat. So this limit of quantitation has both a lower bound and an upper bound, and we need to be able to figure out where that is to be able to quantify effectively. One of the cleverest things I've ever heard came from Dr. Psycho, who explained that mass spectrometry has made us the masters of the x-axis. We're really, really good today in mass spectrometry at pinning down a mass to charge value. M over Z values are where we live, and we get really, really accurate measurements there. We are much less effective at being able to measure intensity variation. I'm able to tell you to within 10 parts per million where on the M/Z axis this ion falls, but my ability to pin down an intensity to even a 10% CV is pretty limited. CV is coefficient of variation. Having measured the same thing many times, we ask what is its standard deviation, and we divide that by the mean. So when you see a 10% CV, that's a measure of something that's pretty good, in analytical chemistry at least, for quantifying a level. So we're not particularly good at getting strong measurements, strong bounds on our intensity variation when we're down in the noise. When you have a peptide that's a very, very minor constituent of a sample, your ability to quantify that well is much reduced. It's far better for you if you're able to quantify that peptide in a context where it's not being overshouted by everything else in the sample. So you can expect a 10% CV to be about the best case of what you're going to get from mass spectrometry quantitation. Lots of takeaway messages today. I've highlighted a few of these along the way, I think. Your quantitative experiment may change depending on whether you need to know simply what changed or how much did it change. Those two have very different requirements for the experiments we run. Label-free methods, isotopically labeled like constituitive labeling or SILAC labeling, isobaric tagging, and targeted quantitation are all legitimate strategies for quantifying. They have different strengths and weaknesses. Generally speaking, I would say that if you want to know that something has changed by a factor of, say, 20 to 50% between cohorts, you're going to be a lot better off with something like targeted quantitation. If you try to repurpose a shotgun data set for the purposes of quantitation, you'll almost always be kind of disappointed by how much variability is associated with that. To get a quantitative result, I strongly urge you to design a quantitative experiment. That is frequently not going to be this broad-ranging shotgun thing, which has so heavily dominated most of proteomics for its entire history. Okay, and with that, I am done talking about targeted quant. I am happy to talk with you on any of the Thursdays that I'm here, and if you have questions about this subject, I'd be glad to set up a meeting with you. Thank you.